Our next session comes from PBS Biotech. PBS Biotech helps manufacturers scale and differentiate cells as final therapeutic products. They work with research and production teams to turn complex bioprocesses into robust, reproducible results. Their focus is always process consistency. Their approach is scalable, linear, and built for the most demanding cell culture conditions. Their focus is always peak production. Today, we will hear the topic CMC by Design, Enabling Process Consistency with Vertical Wheel Bioreactors. Please welcome Sharon Harvey, Production Management and Strategy Director at PBS Biotech. Let's welcome her with a warm applause.
Thank you very much. Appreciate it. All right. Good morning, I'm Sharon Harvey. I'm with PBS Biotech. PBS manufactures a family of single-use bioreactors that are purpose-built for cell therapy product development and manufacturing. And today I'm going to talk about how our vertical wheel bioreactors are changing the way we think about cell therapy manufacturing. And that's by enabling true process consistency. Consistency is what makes your data meaningful at small scale and your process predictable at large scale. And that's where PBS Biotech is focused. We serve as a strategic partner for cell therapy developers as they move from R&D through commercialization. All right, as you know, there's lots of ways that you can expand cells. Most research starts in small-scale 2D, your well plates, your T-flasks, your G-Rex. But as cell yield becomes critical, like it often does with allogeneic therapies, 2D static culture quickly becomes very inefficient, and 3D dynamic platforms become much better suited. And that's because they introduce two key things, and that's scalability and control. But what's most important in this preclinical phase is your expansion platform that's selected is done so with the end in mind. Because the platform you choose early on often defines how and whether you are able to scale it at all. And there can be major cost implications if you make the wrong decision. So hope is not a strategy, guys. Your expansion platform isn't just a tool, it's the entire foundation of your manufacturing strategy. So please choose it carefully. All right, so now I'm going to talk a little bit about what makes vertical wheel different, okay? And it starts with hydrodynamics. Okay, our wheel and vessel, U-shaped vessel, they work together to promote a strong sweeping flow and homogeneous mixing that's ideal for the expansion and differentiation of a variety of cell types. So when users are trying to get from here all the way to there, that wheel to vessel ratio stays the same. That means your hydrodynamics stay the same, and that's going to be critical for enabling linear scalability. And by linear scalability, I mean that as you are increasing your reactor scale, that you're able to maintain the key process parameters, and thereby you're able to maintain those growth kinetics and maintain that same cell density as you scale. And that's going to make for predictable, reproducible, consistent biomanufacturing. Okay, so these particle velocity profiles demonstrate how hydrodynamics differ between stirred tank bioreactors and vertical wheel bioreactors. So stirred tank bioreactors require high power input in order to suspend large particles. And that's going to create pockets of high and low energy within your bioreactor. And that's going to result in shear stress, and it's also going to introduce variability within your culture. Versus vertical wheel bioreactors, they enable controllable power input, okay? So that means that vertical wheel is capable of not only suspending delicate stem cells gently, But you're also able to hone in on the optimal agitation rate that's ideal for your particular cell type. So all your cells get the same treatment over the duration of your cell culture. That's going to improve quality. It's going to reduce variability. All right. But vertical wheel bioreactors deliver clear advantages, not only for shear sensitive cell types like PSCs, MSCs, organoids, your islets, NK cells, but we're also seeing advantages with more shear tolerant cell types like T cells. Okay, and I'll elaborate on some of that data later in the presentation. All right, so here's our growing bioreactor fleet. From R&D through commercial, we've got you covered. Whether you're doing autologous therapy or allogeneic therapy, our portfolio scales up or out with your needs. Okay, but I want to take a beat to focus on where the customer journey starts, and that's going to be with our PBS minis. The minis provide a low-cost, low-risk point of entry for those who might not be as familiar with 3D suspension culture. It's very low-tech, low complexity. It doesn't have all those bells and whistles that you typically see in larger bioreactors. And best of all, it provides a true scale-down model, okay? So that's going to be ideal for your process development and scale-up. But if that didn't sound too good to be true, our minis are single-use. They're qualified GMP clinical grade. The gassing is enabled through the vented caps that are on the top of each of the vessels. The agitation is controlled using the speed adjust dial located on all the universal mini bases. So what you're going to do is you're going to take that vessel, you're going to put it on your mini base, you're going to take that mini base, you're going to put it inside the incubator, and that's how temperature and gassing are controlled for the minis. Okay? Mini comes in two different sizes, the 0.1 and the 0.5. That gives you a working volume between 60 milliliters up to 500 milliliters. And each of those sizes can be used interchangeably on the universal mini base. There we go. All right. We also have a perfusion mini vessel. Okay, so that has a dip tube with a retention filter on the end that's appropriate size for your large particles. So this works for PSC aggregates, it works for MSCs on microcarriers, it may also work for other cell types like organoids or other cell clumps. It does require an external pump to use, but ultimately it provides users with the opportunity to optimize your perfusion strategy in a low-cost, easy-to-use, scale-down model. Okay. All right, so once you've optimized those basic parameters in your PBS Mini, maybe you're ready to use something a little higher octane, we've got something for you. We've got our PBS-MiniPRO. Okay, MiniPRO enables a closed system individual control of up to 24 mini bioreactors. And then once you've de-risked your process at small scale in the MiniPRO, you're able to scale up into our PBS-3 bioreactor. Okay. Now, also exciting, PBS just launched our credit redemption program. So that means that your mini spend converts into credits that can go towards the purchase of a MiniPRO or a PBS-3 bioreactor. So not only are you building a process in your PBS Mini, but you're actually building equity that pays off in future development phases. Okay. All right, a little bit more about our MiniPRO. We're very excited about it. We just launched it in December. It utilizes the same gentle, low shear vessels that you see with our mini bioreactors, but it does have additional ports to enable rapid automated medium exchange. It comes with bioprocess control, so you have your precise inline monitoring and control of agitation, temperature, pH, and gas flow. You can automate from 1 up to 24 mini bioreactors, all using an intuitive user interface. We've got recipe building, remote culture monitoring. This makes it ideal for use for your high throughput DOEs, for allogeneic seed train, scale-up optimization, even autologous cell therapy manufacturing. So here we demonstrate the impact of dissolved oxygen on your PSC culture by comparing MiniPRO to our incubator-based PBS-Mini. Okay, so on the left, you see MiniPRO's, your right, you see your oxygen input profile. So gassing began on day 4, continues to ramp as cell density increases. The DO set point is maintained at 50% and stays consistent throughout the duration of the cell culture. And then on the other side here, you have the growth kinetics. You see that your MiniPRO, that condition continues to grow throughout the culture duration through day 7. But your PBS-Mini, it does start to plateau towards day 7, and that's because it's encountering an oxygen limitation. So MiniPRO is removing that oxygenation bottleneck, and thereby it enables you to achieve higher cell yield. Okay. All right. And here we compare PSC expansion in the MiniPRO versus our 3-liter vertical wheel bioreactor. Now, both systems here were run under feedback-controlled oxygen. You can see they both reached the same approximate density, 2.3 million cells per mil. Those growth curves are right on top of each other. But what's even more exciting is the metabolite data. So you have lactate, glucose, ammonia, and glutamine, which profiles they're tracking right on top of each other. So essentially what that shows is that MiniPRO is enabling the same biological environment of the 3-liter system, and that's going to allow your PD teams to make decisions at small scale with the confidence that they're going to be able to replicate that at pilot scale. All right, so both MiniPRO and PBS-3 generate healthy, uniform PSC aggregates. They have similar morphology across systems. The aggregate size distributions closely overlap. So on day 6, you see a variability between systems of only 18 micron. On day 7, variability between systems of only 11 microns. So to put that into perspective, that's about the size of a single cell. So this level of similarity confirms MiniPRO provides a high-fidelity, predictive scale-down model for PSC aggregate expansion. All right, but we can be used for more than just stem cells. We collaborated with GeneFab and Repligen to create a case study. Here we demonstrate same growth kinetics, linear scalability between the 0.1 and the PBS-3 using fed batch medium exchange strategies. Okay, and then we implemented a perfusion medium exchange strategy, which, as you would expect, demonstrated greater fold expansion in comparison to the fed batch. Most exciting, though, we compared cells that were harvested from the PBS-3 perfusion condition to those harvested from their optimized G-Rex condition, and we saw 2.5 times more relative potency versus that G-Rex condition. All right, but we don't just stop at PBS-3, folks. We got GMP grade PBS-15s as well as GMP grade PBS-80s, okay? And we also have OPCOA and the MiniPRO PBS-3 and PBS-15 that enables seamless centralized real-time monitoring and control. And PBS-80 soon to follow. All right, so far, I've only shown data up to 3 liters, so I wanted to be sure to fit this one in. This data demonstrates pluripotent stem cells, which, by the way, are arguably one of the most challenging cell types to scale. But here we demonstrate linear scalability from our 0.1 scale all the way up through our 80-liter bioreactor in a 50-liter working volume. Okay. So using our vertical wheel and a perfusion feeding strategy, we achieved over 30 billion PSCs in our PBS-15, 200 billion PSCs in our PBS-80 in a 50-liter working volume. And so these results, meaning like achieving this linear scalability from 100 milliliters all the way up to 50-liter working volume, they're unmatched in the field. So we're very proud of this one. And the good news is you can do it too. So we've used CFD modeling to better understand the energy dissipation rates across our family of bioreactors. Once you optimize mixing conditions in your mini scale, you can come to our website. We've got a nifty calculator we built that allows you to enter in your working volumes, then your agitation rate, you press calculate, and that's going to generate what agitation rate you need to use in each subsequent scale of vertical wheel bioreactors in order to maintain that same condition that you saw in mini scale. Okay. All right. And if you're looking for a processing solution for your cell therapy, PBS Biotech has had great experiences using CAR Biosystems devices. CAR uses tubular bowl centrifugation for gentle automated cell processing. So this is going to be critical for maintaining consistent cell quality throughout your development journey and scale-up journey. And they offer 3 different sizes of devices, all in varying throughputs, starting with the UF Mini, then to the Unifuge Pilot. They also have a third one that's even larger, so they can scale along with your process. So you have the UF Mini here, the Unifuge Pilot here. We used the UF Mini. We compared it to a manual centrifugation method. You see that after processing throughout that cell culture duration 6 times, we're able to achieve comparable growth rates, as well as comparable morphology. We also achieved comparable pluripotency. That's not shown here, but you can find that in the case study. It's published on our websites. And then using the known flow rates, we show here how long it would take to process varying volumes using the CAR Biosystems devices. So here you see that the 3-liter can be processed in about 30 minutes using the UF Mini. And then to process 80 liters, it only takes about 100 minutes. And to put that into perspective, there are some processing solutions that it would take over a day to process that volume. So we're quite satisfied with these devices. All right, swinging back to vertical wheel bioreactors, we are manufacturing in alignment with GMP. We also have software that meets our 21 CFR Part 11 compliance. Okay. And this slide shows our vertical wheel bioreactors have been used to advance at least 11 programs through clinical trials around the world. 4 of those programs have scaled up to PBS-15, and not indicated here, but at least 3 of these have gone all as far as phase 3 and are continuing. All right, so in summary, I want to emphasize, hope is not a strategy. Play the long game. Design with the end in mind, okay? Homogeneous hydrodynamics are going to drive your process consistency. And vertical wheel hydrodynamics enable our users to leverage our true predictive scale-down model for early process development. We've also delivered linear scalable performance in 50-liter scale. We also have we support closed automated process control at every scale. You saw our PBS Mini. It goes all the way down to 300 milliliters. And then finally, our bioreactors are built for cell therapy manufacturing with the end in mind. All right, so with that, I'd like to thank you very much for your time. If you haven't yet, we are in the exhibit hall where you can see our PBS3 and our minis. And you're absolutely welcome to reach out to us at sales@pbsbiotech.com. All right? Thank you very much. Any questions? Yes?
Hi, I'm Elia Sanchez from Biomarine Pharmaceutical. I'm curious about the chemical composition of the bioreactor itself. Is it glass? Is it PPE? And what is the validation portion of the bioreactor? You know, because in stainless steel, we we use clean in place and all sorts of challenges to make sure there's no accumulation of any bacteria or fungi. So what's that part? Yeah.
I don't know the specific plastic type. It is plastic. And we qualify our vessels with various safety testing to ensure that it's appropriate for use in clinical. Mm-hmm.